Hello everyone, I am Yanlei Liu. I will introduce a paper published in iMeta journal What Determines Plant Species Diversity Along the Modern Silk Road in the East. As we all know, environmental samples contain a lot of genetic information in the form of eDNA. Plant information is also included. However, until now, the focus is still on microorganisms and animals, although some review articles have introduced the discussion of using environmental samples for plant diversity research. No research results have emerged. In fact, we think that plants are more suitable for such research. The DNA in soil samples can be specifically enriched with chloroplast-related gene fragments, and the plant species and relative abundance can be obtained through high-throughput sequencing and bioinformatics analysis. Compared with animals and microorganisms, the species classification of plants is relatively perfect. In China, Flora of China, China Higher Plant DNA Barcode Reference Database have been built and the available chloroplast-related data is rich, which is to meet the requirements of using environmental DNA for plant species diversity-related research. Moreover, current research focuses on the red areas with high species diversity. We regard the area in the blue circle as the cold spot area of species diversity. These areas generally have few plant species and are more sensitive to environmental changes and human activities. Now the world is in a period of expansion of cold spots and contraction of hot spots. The main reason is that desertification is rampant in the world. Therefore, the research on plant species diversity in cold spots is conducive to desertification control. Therefore, we chose the desert area in northwest China, which is the nearest and easiest to realize, as our research area. This region belongs to the eastern part of the Asian inland desert area, and is the only land route of the ancient and modern Silk Road. The population density of different regions in this region varies greatly, making it easier to find the impact of human activities on the local plant species diversity. We collected 144 surface soil samples along the northern line of the ancient Silk Road in this area. The sample collection area is wide, with latitude spanning more than 5 degrees, longitude spanning more than 30 degrees. The total DNA in soil environmental samples was obtained by the total DNA acquisition method, and then the target fragment barcodes were amplified by PCR using the commonly used plant chloroplast DNA barcodes. The amplified amplicons were constructed and sequenced in a high-throughput data library, and then the subsequent data were analyzed using bioinformatics methods. Finally, this study found a total of 671 plant species in this region. Angiosperms account for 97.8% of the total. Fabaceae, Asteraceae, Rosaceae, Poaceae, Amarantaceae are the families with a large number of species. By integrating the plant species information of each point, the plant species diversity in this area increases first from the east to the west and then decreases. This trend is different from the pattern of plant diversity in our subconscious. Through further analysis of the sampling points, we found that the first 20 samples in the east are in desert areas, and the vegetation in these areas is very rare. After removing these samples, this area presents a pattern of high plant species diversity in the east and low in the west. The results of correlation analysis showed that the diversity indexes of local plant species were negatively correlated with temperature-related factors. It is positively correlated with longitude and altitude. Longitude is positively correlated with plant species diversity. Temperature has become the main factor affecting local plant species diversity. There is no significant correlation between precipitation and local plant species diversity. We speculate that this may be due to the fact that local plants have adapted to scarce precipitation for a long time and gradually turned to absorbing the groundwater melted by glaciers as the main source of water demand. We named it groundwater dependence hypothesis of plants in arid areas. However, this theory still needs further data validation. Based on the cluster analysis of species diversity among the sample points, combined with the correlation analysis between the main human settlements and plant species diversity and population density, this study found that the plant species diversity is low in areas with large population density, which indicates that the intensification of human activities in the northwest desert region has reduced the local plant species diversity. Finally, we proved that environmental DNA is indeed an indicator for rapid and accurate assessment of plant species diversity. Combined with label PCR, it has become a reality to use environmental samples to study their species diversity with limited financial support. This is also the first large-scale study on plant species diversity using environmental DNA so far. In the northwest desert area, temperature and human activities have a great impact on the local plant species diversity. We have put forward the hypothesis that vegetation in desert areas depends on groundwater, and will design experiments to verify in the future. This is the main research content of this article. Thanks for listening.